Good afternoon, ladies and gentlemen. I apologize for the slight delay that's occurring because the slides haven't got loaded. And welcome you all, my earlier CEOs and doctors, friends of mine who've been here. So probably I'm one of the clinicians who's going to talk about something very close to the heart of many doctors and many administrators, the difference between the original and the generics, and whether this really creates any kind of difference in outcomes, especially so in the ICU. So this came back way back in 2008, and they talked about Jan Aushadhi, accessibility, affordability, and acceptability to start using generic medications. And somewhere in 2012, this guy came up. He came up in 2012 and stirred the honest nest, and he tried to get generics done. He cried his way to millions of dollars, millions of rupees. He did, and he did talk some things that are right and some things that's probably not very right in medicine, is what I felt. That's my opinion. But what the Medical Council of India did do in its Medical Code of Ethics, they put up this statement, a very, very powerful statement. Where is a powerful statement that says, every physician should, as far as possible, prescribe drugs with generic names, and he shall ensure that there is rational prescription and use of drugs. So, ladies and gentlemen, generic branded drugs, does it impact outcome? This is what I'm here for. And I have no conflicts of interest for this talk. What I'm going to do is, through this presentation, the next 15 minutes, I'm going to look at how we move from original to generic to bioequivalence, a little bit of animal outcome studies, a little bit of human studies, and probably summarize the whole thing with my view. When we look at primary health centers and community health centers, the basic bulk of cost of healthcare, apart from resources, is on drugs. And this is from public healthcare, that is PHCs and CHCs 2 years back in India, which actually looked at the red bar being showing the amount of healthcare expenditure on drugs alone. Well, let's see, when we write a drug, the CEO feels, aha, I know why he's writing this drug. Probably he's getting to a junkets. When the drug comes to the department, we see there's something else coming in, and then we start mentioning, oh my God, this is why it's coming in, because he's probably got the margins. I mean, why does this come? This is probably because of the huge amount of difference in the margins that we get, huge amount of difference in the cost of the drugs between the originals and the copies. So why are these originals so expensive? That's the problem. That's the real problem that we must understand. So market forecast clearly mentions that it is going to be the generics that are going to go up. It's very, very clear that by year 2017, we already see 90% growth of generic markets, and the maximum market is from India. That's what it looks like. The dark blue mark, so we can see over there, is the maximum amount of market is from India, and that's why India is called the pharmacy capital of the world. So why are these original molecules so expensive? Why are these original innovator brands so expensive? Well, it comes at a cost because, A, what do they need to do? They need to first get an investigational new drug approval that goes after animal studies. After doing a large number of animal studies, they need to go ahead and they need to approach the Food and Drug Administration to ensure whether they can go ahead and start testing this kind of a molecule. So among 20 to 60 healthy volunteers, they are tested very well on them for side effects, and then this is gone ahead and tested for around 300 to 400 patients to figure out about outcome and clinical effectiveness, and then scaled higher to somewhere around 3,000 patients, so that it can be then taken ahead to market into the departments. But then what happens after this is that they take up all these studies, they analyze them, make a file, and then they send it for an NDA period, that is a new drug approval period, followed by a new drug approval period, which then goes ahead and the FDA then goes ahead and makes a regulatory authority, a body that scrutinizes these studies. They sit and they scrutinize these studies, and then they have a couple of checks in the factories, and that's when the innovator drug comes out. So it is involving a huge amount of research, a huge amount of paperwork, a huge amount of cost and expenditure that then relates to the formation of this drug, and that's the reason that probably these drugs are very expensive. The innovator molecules or the original molecules are very, very expensive. What are generics? Well, generics are basically first copies. I mean, they're copies of the original molecules, but they have to prove bioequivalence. They have to prove stability, potency, purity, and they have to follow good manufacturing practices. That is for certain that these need to do. Now, what is bioequivalence? It means you give a drug into the patient, then there is a curve that comes inside the patient, a Cmax, an area under the curve, which actually tells the extent of absorption, and then a Cmax as to how quickly it reaches the maximum concentration, which then corresponds to extent and rate of absorption. So that's what the bioequivalence is all about. It's a laboratory model that looks at this. And the new drug that is made, the generic that is made based on the original, should be somewhere between 80 to 1.25. That's how it looks like, zero point 0.8 to 1.25. So if the generic lies somewhere between here, then probably it's ready. It's ready for use, and this can be given approval for use in marketing. Well, that's why it's good enough. But these are for oral drugs. This may not be the case for IV drugs, is what it looks like. So are there evidence that our drugs that we are using in generic are equivalent or rather better, or at least does not cause side effects with original molecules? Well, look at this study. Enoxaparin, very, very frequently used drug. So this is a bioequivalence study for enoxaparin with generic molecules. And they looked at 22 subjects. They looked at AUC, Tmax, Cmax, the usual bioequivalence, what is required for. And what did they find out? The results are that they are bioequivalent and they are tolerant with no side effects at all. I mean, this is what it looked at. They even looked at anti-10A, which is the marker with which you figure out whether Clexane works or not. Well, there are so many studies across the board that comes in to talk about bioequivalence, about generic molecules and innovative molecules, that it actually dispels the doubt with respect to bioequivalence. Now, bioequivalence is a laboratory model. So when I Googled up bioequivalence, I found out around 700 odd studies in the world, and out of which 60 studies were there from India, which talked about bioequivalence. Well, this is one study that came in 2011, Indian Journal of Pharmacology. They looked at two different brands of ciprofloxacin. And what did they figure out? They looked by high-performance liquid chromatography that they are absolutely the same with respect to weight, dissolution, particle size, and absorbability. This is what they looked at. Another study in 2017 comes in. Now, what happens in 2017? They looked at a very, very, very highly used molecule called as amoxicillin. They used that amoxicillin, and they compared that to the generic molecules. That means the test versus the generic molecules. And what they figured out was there was a huge amount of variation in AUC and MIC levels when they looked at amoxicillin with a certain generic molecule. In fact, the time above MIC, AUC above the MIC, the area under the curve above the MIC, for the generic was so low that it might have affected clinical and bacteriological cure. This is what it looks like. Not nice, definitely not nice. So what did 2005, what did Nightingale do in 2005? They looked at 65 generic clarithromycins, all right? They looked at it manufactured in 18 countries, of which 50% of those were from India. And what they found out that among 65 of those, 6 had less amount of clarithromycin than it actually should have had. They found out that 17 of them were low releases. They didn't release that amount of clarithromycin that's required at 30 minutes. They found out that 12 out of 65 exceeded total impurity limits. And they figured out that 1 in 65 had a problem with dissolution. So there were problems, definitely. There were problems. There's no doubt about it. But does they cause any effect? I mean, they are causing some problem within the laboratory, but are they causing any clinical effect? So looking at that, they try to predict the clinical efficacy via Monte Carlo simulation model, and they looked at 34 generics. And what they found out that most generics, again, failed to achieve the pharmacokinetic and pharmacodynamic parameters. This is what it looks like. So there may be a risk for microbiological failure as per a simulation model. Now, what happens otherwise in vivo? So they looked at laboratory, they looked at mouse thigh models. And they looked at all these various molecules that are there, looked at oxacillin and the various grands. So what they found out was the black mark down there is the innovator brand. It dropped the bacteriology quickly as compared to the other molecules. There is some problem. Does this relate clinically also? That's the question. So when you looked at generic versus originals in ceftriaxone, yes, in clarithromycin, it did not cause any change. It did not cause clinically in human volunteers. That was thigh mouse model, that was laboratory. In human volunteers, it did not show anything. Another retrospective cohort actually shows that it does not cause any problem. This is what it looks like. Concluding, similarly effectiveness, well tolerated, and no significant adverse effects. So 1 month back, a study came out from the US. They looked at such a huge amount of millions of populations, 39.5 million people. They looked at it whether to prove that generics were less effective or more effective. What did they do? They looked at 3.5 million patients. What did they find? They found out that there was no difference in outcomes. I mean, 3.5 million people, 1 month back study talking about that it doesn't cause much of an outcome study. What happens with there is 1 or 2 studies that talk about small decrease in effectiveness. This is a study that comes from Greece that shows that post-operative outcomes weren't good when you use generics. So they're affordable prices. Are we really killing our patients by doing these generic molecules is the question that comes in. Or are we doing anything different? So what happens when you use molecules? You look at the graph, what happens over there is as we use the molecules, as we use these generic molecules, the cost is definitely going down, the usage is definitely going up, and that is relating to resistance. That's another problem that's coming into generic molecules. So what do guidelines say? At least epilepsy guidelines says don't use generic molecules. Industry supplies data about safety and efficacy of drugs. Doctor prescribes the generics. Will it or won't it work? It's basically clinical gestalt. It depends on quality checks. Look at Kerala. Kerala, 6,000 drug samples tested per year. Only 3 lakhs, but 3 lakh samples need to be tested. So the testing was too less. But don't worry, it's definitely improving. Quality checks are going up. The problem is jugaad. This is a word that is put in the Oxford Dictionary. We all do a lot of jugaad. That's the problem. This jugaad should not be the way we treat quality checks. It has to be done the right way. And that's why when you ask a pharmacist, should we or shouldn't we use generic molecules? It looks like if you ask him, do you personally buy generic medicine instead of branded? He says, no, I buy branded drugs. So what do? Well, I feel there's no problem. I've been using it for the last 7 years, and there isn't any problem for generic molecules is what I feel. So there should be regulatory changes on testing generics to instill the kind of faith in generic molecules. There has to be raw material sourcing also data available. There has to be strict quality control, and there has to be state-of-the-art PKPD is what I feel. As of now, it is clinical gestalt, and hence it makes a lot of sense to ask the clinician what he's going to do and look at the outcomes and then decide whether he should use a generic or an original molecule. Thank you very much.